Since fall 2020, Bryce Mericka, an agriculture student at the illustrious North Carolina A&T University, has been dedicated to his research project to find the collard and kale varieties that show resistance and susceptibility to the Harlequin bug, scientifically known as Murgantia histrionica. They are southern bugs, usually found in the Atlantic to the Pacific coast. Collard varieties used in this experiment include Old Timey Blue, Sexy Mama, Yellow Cabbage, Alabama Blue, Vate, and Cascade Glaze. Kale varieties used were Black Magic, Scarlet, Red Ursa, Red Russian, Dwarf Siberian Improved, and Dwarf Blue Curled. Measurements were recorded over the course of a year to observe the changes, if any, between fall and spring. There are various components and factors needed for the successfulness of this project, so I encourage you to listen. First, we grow the collard and kale varieties that will be transported to the greenhouse for care, then to the lab. In the field, there are 4 blocks in which 6 rows of collards, each of a different variety, are followed by 15 rows of kale, also each of a different variety, in each block. The plot itself is 36 meters wide by 40 meters long. Each row is 8 meters long and contains 20 plants, all spaced 15 inches apart. A control section of 4 meters was utilized, which was derived from a buffer of 2 meters at the front and back of each row. Here you can see the high population of the Harlequin bug present on the different kale and collard varieties and the different damage that was caused by them. It also must be noted that the higher the population of the Harlequin bug, the higher the damage. For the harvest data collection, a 4-meter control section was studied where 5 random plants were observed as only marketable, viable leaves were harvested and tagged with pink ribbon. After the leaves were placed in a brown paper bag, the plants were weighed. On the observed plants, 3 to 4 young, developing leaves were left on each plant to ensure growth of the harvest plants. For sugar data collection, to collect the sugar content, 5 healthy leaves are taken off of 5 different plants in the 4-meter control section and placed in a plastic bag after being measured for harvest quality and size. Next, the leaves are grinded to produce a juice that is squeezed for a sample for the digital refractometer. To prevent cross-contamination, the refractometer is cleaned with deionized water in a microfiber cloth. For the chlorophyll data collection, a SPAD 502 meter was used bi-weekly during field observations to collect leaf chlorophyll content by measuring the amount of light that is transmitted through the sampled leaf in a non-destructive manner for each variety of collard and kale. 3 leaves from 3 different plants of the same variety were measured for chlorophyll content, totaling 9 observations for each collard and kale. Leaving the field and into the lab in a 60 by 60 by 60 BioEquip unique collapsible cage, 30 to 45 day-old collard and kale varieties are used versus the control Georgia Southern collard. 2 cages are being used to observe the damage caused by the Harlequin bug. 1 cage holds a single potted plant of each of the 16 collard varieties, including the control Georgia Southern. They are placed randomly into the cage in a 4x4 orientation. For kale, a single potted plant of each of the kale varieties and 1 potted plant of the Georgia Southern collard are placed into the cage in a 4x4 orientation. 50 adult Harlequin bugs, male and female, are starved for 24 hours before inoculation into the potted plants in the cage using a Crayola wash brush. In this test, the arena is arranged with 2 layers of paper towel divided into 4 quadrants and moistened with water to preserve plant segments from wilting during the duration of the study. 2 leaf discs of each collard variety measuring at 4 centimeters diameter are placed in opposite quadrants, and in the alternating quadrants are the Georgia Southern collard leaf discs. 4 adult Harlequin bugs are placed into the arena and able to choose between the different substrates. The damage was recorded every 24 hours for 3 days and scored on a scale of 1 to 5, 1 being little to no damage and 5 being 100% damaged. The growth and development test is essentially a no-choice test in which second instar nymphs are placed into a container and reared until reaching the adult stage. Days to maturity, time between each instar phase, and mortality can be indicators of antibiosis as a mechanism of resistance. There have been multiple versions of data collected for this experiment, as I have discussed. In this graph, you are able to observe the infestation affected by the collars and kills by the different stages of Harlequin bug. Though the data analysis has not been completed, the data collected thus far shows an interesting separation in resistance and susceptibility to Harlequin bug feeding damage and oviposition amongst the varieties being screened. Collar varieties such as variegated, event star landrace, and even our control, Georgia Southern, have been observed to be more susceptible to the Harlequin bug damage. The collars believed to express resistance are the Cascade Glaze CAS, which had low yielding but low infestation and subsequent feeding damage. Yola cabbage collars also experienced low yielding but fairly low infestation and damage rating. There was also significantly different chlorophyll content. The kale varieties believed to express resistance are all of the green and blue-green curly kale varieties that posted lower feeding damage ratings, such as dwarf blue curled Fates (DBCV), dwarf blue curled Scotch (DBCS), winter boar (WIN), Bates blue curled VBC, dwarf blue curved DBC, alongside lower infestation numbers. STN F12 hybrid kale had the lowest damage rating, which was significantly different from other varieties. STA was also atop the highest yielding curly varieties. Bryce has completed this experiment with hearty dedication because that's what Aggies do. Thank you.